Good afternoon, everyone. My name is Paige Mackenzie, and I'm a first-year master's student at UCT. I'm doing my master's in medical biochemistry. Every 10 minutes, a South African dies from TB. And even though the efforts to reduce TB deaths have increased, the number of the shocking statistics like this remain our reality. Structural biology in TB is an important part of research. I'm sorry, let me start again. Sorry. Okay, so it is also important to note that the number of effective and available broad-spectrum antibiotics available is steadily decreasing, and the time and effort needed to develop new antibiotics consistently thwarting the efforts to develop new ones. It is no secret to those of us in this room that structural biology is an essential component of biomedical science. Even globally, there has been a push to solve all proteins implicated in human disease, and MTB research has not been left behind. Pre-pandemic, post-pandemic, we have more than 7 times the amount of information that we did in structural information that we did pre-pandemic. What is shocking is that this actually only represents 10% of the available MTB proteins expressed. UCT, the EMU at UCT is the only facility in Africa with the capacity to undertake high-resolution experiments, and the MMRU is a lab that is a leading force in MTB research. And so this means that UCT has not been left behind in the structural research of MTB. And Reese Wilmore, a previous master's student at UCT, reconstructed encapsulin, which is a protein in the membrane of MTB. And during my honors year, when I was learning how to do cryo-EM for the very first time, we noticed that one of Reese's data sets had these pilin-like structures or filamentous-like proteins in the data set. And so during my honors year, which Jeremy included in his slides, I attempted to reconstruct this protein in a very short amount of time. And at the beginning of this year, tried again and succeeded a little bit more to get a low resolution but pilin-like structure. And I thought this was very interesting. And so what are bacterial pilin? They are long rope-like appendages that are expressed on the cell surface of bacteria, and they span both the inner and outer membrane, or just or expressed from the outer membrane. And they are involved in a number of functions in a variety of bacteria and archaea. And in pathogenic bacteria, these include DNA uptake, phage transduction, and a form of bacterial locomotion known as twitching motility. Additionally, they also form as cell adhesion proteins, in which it is the first point of contact, really, for a pathogenic bacteria to interact with its host cell. In gram-negative bacteria, the pilin have been largely researched and reviewed, and they are categorized and subcategorized by their biogenesis and assembly pathways. In gram-positive bacteria, they've been less researched, but are also less variable than gram-negative bacteria, and they tend to form by covalent polymerization. Similarly to MTB research in structural biology, there has been a growing interest in the structural characterization of various bacterial pilin. And this is because they have a really interesting potential for therapeutics or vaccine design. They are not found in human cells, and they are not under the protection of a bacterial capsule or membrane, and are thus vulnerable to attack. And so, additionally, antibacterials that prevent virulence have also shown to be more effective in preventing the emergence of antibacterial resistance, and thus the interest in antimicrobials that mechanically disrupt the pilin have been researched. And so there are pillicides that have been designed against uropathogenic E. coli. And what is interesting is that these did prevent virulence, and another 2 different types of pillicides prevented virulence against more than one type of pilin, which is very exciting. And so I was interested, obviously, in Mycobacterium tuberculosis, and I wanted to see if the filament-like structure that we saw in the beginning negative stain images were pili. And so a short literature search showed that Mycobacterium tuberculosis did produce pilin, and these were characterized as the curly type pilin and the type 4B pilin. What is interesting is that the research that was spurred by this initial finding by Altieri et al. In 2005 found that there were contentious results, and that the definition of the pili type and the characterization of pilin as a virulence factor were different. And so it is also worth noting that the sequence homology between the open reading frame that is thought to produce the bacterial pilin and other bacterial pilin is very low when we did a blast search against them. The only thing that produced some kind of sequence homology was that the MTB protein had a 74% sequence homology with MSMEG. And so an interesting piece of recent research is that Terwilliger et al. Showed that you can use the atomic map from unidentified protein complexes to identify protein identity. And this is an important point for the next part of my master's project. And so this eventually brings me to a brief outline of my project, and the aim of my research is to use cryo-EM to identify and clearly define the structure of a bacterial adhesion protein, or pilin, produced by MTB. The significance of this work lies in the use of an innovative and advanced technology, cryo-EM, to investigate a classical virulence factor present on the MTB cell wall. And so I'm going to now quickly run you through some of my initial results that I've got in the first couple months of my master's. And the first thing we did was just to see if MTB did in fact produce these pilin. And what we did was an approach commonly used by Ed Eagleman, where you simply inoculate the bacterial culture and then incubate this bacterial culture on a carbon-coated grid, and then view the bacterial culture on by negative stain. And this is essentially what we did. And unfortunately, there was quite a lot of contamination. So we wanted clean images, and that's unfortunately not what we got. But there were these promising outgrowths, or kind of dense staining on the exterior of the bacterial cells that was fairly similar to what we'd seen in Alteri's publications. And so we kind of carried on. But I chose to optimize a pilin purification technique in smegmatis so that we could find a way of looking at the pilin more closely. And so the first question was how we were going to get this pilin off of the bacterial cell surface. And it turns out that just vigorously vortexing them for an extended period of time manages to shear the pilin off of the cell wall surface. And then concentrating them with a spin concentrator has increased their presence on the grids. Interestingly, ammonium sulfate precipitation caused an extensive amount of clumping, and that hasn't we've decided not to move on with ammonium sulfate precipitation. Something worth noting is that quite recently there was a publication on a type 1 pilus structure from E. coli, and in just observing the ultrastructure of both of these proteins, they look remarkably similar. It is worth noting that Alteri, according to the sequence homology, classified this pilin that we're interested in as a type 4. And so using Terwilliger's approach of using the atomic map could be quite an interesting way of clearly defining that pilin type. And so what next? I'm very excited. I've got a high resolution cryo-EM dataset collection at EBIC, hopefully in the middle of September, if I can get my sample prepared in time. And in summary, I'm going to be using automatic map interpretation from 3D reconstructions of mycobacterial proteins to generate an atomic model that will facilitate the identification of these proteins by matching them to their corresponding sequences. And second of all, my cryo-EM reconstruction will be used to investigate the value of mycobacterial pilin as a target for new anti-TB drugs and vaccines. I just like to take a little bit of time to thank the CZI Fund for funding my training, and the various labs for facilitating my lab work, and to Vanessa Samuels for teaching me how to culture bacteria, and for Jeremy for helping me learn the ins and outs of cryo-EM reconstructions. Thank you.
Thank you, Paige. That was very interesting. I have one question before I pass over to everybody else. Did you do an AlphaFold 3 prediction of the structure of the monomer?
So we did. The AlphaFold 3 structure, obviously when you do a monomer, it didn't look like a helical filament at all, because it was just this very small 6 kilodalton protein. And then when I made it into a polymeric or a multi-protein pilin, the pilin structure was very strange, and I think, and it didn't form this helical filament with strong symmetry or rotational symmetry. And I think this is because AlphaFold 3 and generally AI use homology modeling as their main basis for reconstruction. And when I did the sequence homology against my protein to other pilin types was very low. So I think that might be why it was very strange.
So did you do it with multiple copies?
Yes, yeah, the monomer, yes, to build a helicus.
All my attempts to use AlphaFold 3 predictions on multiple monomers have been laughable.
Yes.
So, okay. Any other questions? Any? Yes. You better hold this one.
Thank you.
I'll get it to people.
So thanks for a really interesting talk. You highlighted that this question of whether mycobacteria actually produce pili is quite controversial, and it's indicated in the list of references that you've got up there. I was wondering if there's a way, because just doing a quick scan of the literature, where people have basically tried to identify the gene that is involved in pilin biosynthesis, and then knocked it out and shown under EM that there's an apparent absence of pili, but your results have already highlighted how difficult it is or can be to find them in the first place. So that's always felt like a very inconclusive demonstration of the presence of pilin.
Yes.
So I was wondering if there's anything you can learn from other bacteria, if there are mutations which can be tolerated and change the pili structure in a way, in a very obvious way, and that those could be engineered into the mycobacteria to demonstrate that what is actually producing the pili is that gene, and making that that link between the producing gene and the final structure much clearer. And I think that would aid in your own research as well, in convincing the naysayers that who argue that this is a figment of people's imagination. Yeah.
Yes. I just an interesting, we do, Jeremy might have to correct me on this, but we do actually have access to a MTB strain that has got every gene knocked out. Am I correct? Yes, we've got a gene library. Sorry, not a.
So this is not necessarily not that you want to overexpress it.
Yes. So overexpressing that bacterial pilin in a recombinant, the gene that we think is the pilin in a mycobacterium smegmatis might show us that by overexpressing it that they're more, and therefore that this is the gene that is producing these pilin. Additionally, the paper that was recently published on the type 1 pilin, these are dynamic machines. They have ATPases that facilitate the extension and retraction of the pilin, and they've shown that if you delete one of the two ATPases that function in this molecular mechanism, that the pilin is actually incapable of forming a long, stable filament structure. And it would be interesting to see if we could delete a similar ATPase in one of those bacteria and in mycobacterium tuberculosis, and to see if that could perhaps change the elongation. Because in our experiments, the filaments are very, very long. And when the researchers have done pilin investigations in recombinant E. coli with the MTB gene inserted in that E. coli, the pilin are much, much shorter. So it'd be quite interesting. And then additionally, they have shown that the gene expression profile of this open reading frame increases when the bacteria comes into contact or enters the macrophage. And so if I can prove that using the Terwilliger, that the amino acid sequence sequence is truly this sequence, and that the that gene expression increases upon interaction with the macrophage, then be quite an interesting result.
Thank you very much, Paige.